Our second speaker of the session. The second speaker is Professor Dr. José Sampaio. I hope I pronounced your name correctly. From the Applied Biomolecular Sciences Unit, Department of Life Sciences, Faculty of Science and Technology, Universidade Nova de Lisboa, Portugal. So your talk is going to be traveling through the unseen biodiversity, a personal account. The floor is yours.
Thank you, Professor Repi, for the introduction. Do you hear me? I'm okay? And also thank you the organizers for inviting me to be here today to talk about a topic that closer like this. Okay. To talk about a topic that has been already mentioned, but just briefly. Well, in the previous talk, we heard about small animals. They are extremely important. So it appears that size is not important when we talk about biodiversity. And I want to move even further in the scale of small things. I would like to talk about microbes. And why is it important to mention microbes in a biodiversity conference? Well, because our entire lives depend on microbes. They are essential for catalyzing all environmental transformations for which we depend. Earth is 4.6 billion years of age, and this is an enormous, so it is a number that we cannot grasp. We cannot grasp millions of years. But we can do a simple trick. We can compress the entire lifespan of Earth, of our planet, in a single day. So imagine that Earth was formed at midnight. Well, by 4 a.m., life was already thriving on our planet. Of course, this was microscopic life. And then what happened at 6 a.m.? Well, again, only microbes. And at 8 a.m., well, microbes. Noon, microbes. What happened around 5 to 6 p.m.? Well, we see appearing the first multicellular organisms. Are they visible? No, they still are invisible. So the things that we have been discussing here, plants and animals, so this starts at 9 p.m. Mammals, 20 minutes before midnight. We humans, 1 minute, 17 seconds in the history of our planet. So our planet, if we talk about life, we need to talk about microbe life. Earth's biosphere is completely shaped by what microbes do and have done since the beginning of life. Think just on oxygen. Where does it come from? So there is a microscopic, invisible, but majority of biodiversity that we need to consider if we really want to discuss biodiversity. A few examples. Marine phytoplankton, so the marine microbes. What do? Well, half of the global photosynthetic CO2 fixation is done by microbes. This is global numbers. And half of the oxygen production in our planet is done by microbes. Prokaryotes, which are bacteria and archaea, together with fungi, they represent 15% of Earth's total biomass. What do animals represent? Well, less than 0.4%. What about oceans? 90% of marine biomass is microbial. What do? Well, they fix carbon and nitrogen. Like no other life forms. do the opposite. They remineralize organic matter, and they are therefore the basis for all the ocean food webs. So the scientific community is gradually becoming aware of the relevance of microorganisms for such important problems as climate change. So this is a document by many researchers that raises awareness on the importance of microbes for climate change. So now we have a link, and this paper discusses this extensively, between microbes, plants and animals, and climate change. How are microbes being affected by climate change? We don't know very much about this. We have a few hints, but we don't know very much. Now, I was invited here to talk about international collaborations related to biodiversity, and I will be talking about fungal biodiversity. And here, so I am coming from Europe, from Portugal, and here, we are here. And for several years now, I have been collaborating with Professor Savitri Limtong, and also more recently with Professor Nantana Srisuk, and also with Dr. Sasitorn Jindamorakot on the topic of microbe biodiversity. What do we still do not know about microbe biodiversity? Well, many things. Most microbial species, we don't know them, and many of them, we know them only indirectly. We are not able to cultivate them. What do I want to know? Well, mostly I want to know about microbial ecology. I want to know what are the microbial niches. In the same way, we know about the niches of plant species and animal species. We don't know this about microbes. We have some tremendously important tools right now that are based on genomics, or the analysis of the complete genome sequence of the different microbes. We can recognize better than any time before, we can recognize how species are organized, and we can even understand populations within species. With Ana Pontes, a student in our lab in Portugal, she did a map of populations of a yeast species, the most famous superstar yeast, Saccharomyces cerevisiae. Why is it important? Well, because it makes bread for us, makes wine, makes beer, it makes sake, and makes many other important fermentations. So, we are trying to understand how the species is organized in terms of populations. And we want to address these questions that I already mentioned using these species as a model. Because we know very well the origin of each of our samples that we are sequenced the genome, then we can correlate their position in this phylogeny, so the groups that they form, with them being wild or domesticated. So now we can have a map of multiple wild populations and also multiple domesticated populations. So this is not a work by ourselves. Other groups have been working on this topic. So now we know that the species, this microbe species, has a wild, natural history, and also from here we have obtained what we call domesticated populations. Why do we use this name domesticated populations? Well, because microbes have been domesticated more or less in the same way as we have domesticated plants like corn or animals. We have markers of domestication that we can identify in their genomes. So understanding the natural biology, of course, requires fieldwork, and in Southern Europe, we could associate the isolation of these yeasts from oak trees, especially the bark of certain species of oak trees. So this is not a very normal or This is not an expected habitat for a yeast that likes to live on sugar and so on, but this is the results that we obtained. Now, going back here to Thailand, with our collaborations, we could identify novel populations here and here, found so far only in Thailand, that have some peculiarities. And I'm going to talk just briefly about a group of genes that is necessary for the assimilation of galactose. So I will go really briefly, just highlighting an important aspect, not the entire study. But imagine that you offer two sugars, glucose and galactose, to a yeast that is growing on your test tube. So what happens normally is that glucose, which is this red line, is consumed first. So this is a glucose lover yeast. It uses first glucose and has processes of repressing the enzymes for consuming other sugars. Only when glucose is exhausted, then we start to see the consumption of galactose, which is here in green. So, okay, this is the normal pattern. So now let's take a look at this Thai population here. Okay, the red is here, so it's consuming glucose, but before glucose is totally consumed, galactose starts to be consuming. So in these populations, they have a different process of organizing sugar consumption. They still prioritize glucose consumption, but they are less repressed. So why is this happening, and what is the ecological basis of this phenomenon? Well, galactose is the sugar that results from the degradation of lactose, which is the main sugar in milk. But of course, here, this is a very different habitat from milk, and these are wild populations. Well, there are other molecules that also have galactose. So this is the first one here. Okay, the second one. This is raffinose. Raffinose has this part, which is glucose and fructose. This is called sucrose, this combination. And then it has one galactose residue. So this is raffinose. Then this other molecule, stachyose, it has the same structure, but two galactose residues. And the third one has 3 galactose residues, and the fourth one has 4, and then there are even more complex molecules. But the thing is that they are packed with glucose. And where do these molecules occur? Well, in plant material. So now we find an association between this better ability of using galactose and an adaptation to a natural environment. We are getting closer to the microbe ecological niche. Okay, international collaborations. They are ruled by protocols, they are ruled by regulations, and one of the most important regulations is the Nagoya Protocol that attempts to give access and benefit sharing to those countries that, let's say that they harbor the higher biodiversity, they are less explored in terms of knowledge, and now this protocol attempts to give these countries access to potential benefits, especially economic benefits. This is, of course, something that it is difficult to argue against, but this protocol was thought in terms of plants and animals and not in terms of microorganisms. I do not go into details, but there are, if you want, you can read what it is here, but there are specific problems associated with microbes that are not covered by this protocol and that get scientists to be very confused. And the consequences of this, so this protocol started to be implemented in 2014. The confusion is so high among scientists that it is contributing to paralysis. It is contributing to abolish international collaborations. Now, collaborations, if they are true, they go both ways. So it's not me coming to Thailand, it's Thai students, Thai young students coming to Portugal to learn about these important genomic techniques. Why? Because then they can come back, they can implement these techniques here. So this is what I have to say. My take-home messages are very simple. Microbes are very important for biodiversity. We need to know much more about the ecology of microbes, and international collaborations will allow us to do this, especially if they are two-way collaborations that allow for capacity building. So this is my people in Portugal, including my two Thai students. One of them is already here. Varunja presented the talk earlier this afternoon, and I would also to acknowledge the funding that we have. And thank you for listening.